Good.
Thank you for the invitation to talk to the committee today. I'm going to go over the regulation of agricultural biotechnology products that we do in APHIS and specifically in the Biotechnology Regulatory Service. Some of what I'm going to say has actually already been covered by Sarah and or Larissa, so hopefully that will help me speed through the presentation here, and I'll just kind of refer back to what some of the things they've said. Sarah already gave an overview of the coordinated framework, and this slide just kind of goes over that again. I won't spend a lot of time on this because Sarah has mentioned it. Under the coordinated framework and particularly for genetically engineered plants, as Sarah mentioned, there's 3 regulatory agencies that are involved, USDA, FDA, and EPA. And I'll talk today about USDA and our work to ensure that GE products are safe for use in agriculture. Our regulatory authority comes from the Plant Protection Act of 2000, which actually provides, the slide says 2 authorities, but in fact there's 3 authorities. We haven't really used third authority yet in any way. But there's 3 authorities that could be used in regulating GE organisms. First is the Plant Pest Authority, and that's the basis of our current regulations. That's 7 CFR Part 340. As Sarah mentioned, there's also a Noxious Weed Authority, and we proposed in our 2008 draft rule to take on the Noxious Weed Authority, and taking on that authority remains under consideration as we are moving forward with revising our regulations. And then third, and not shown on the slide, is the Biological Control Organisms Authority, which Sarah mentioned. And we haven't had any genetically engineered biological control organisms yet, so that hasn't until recently entered much into our thinking, but I think with the emergence of gene drive technologies that may certainly enter more into our thinking. What we regulate is defined in 7 CFR Part 340 by the term regulated articles, and regulated articles are essentially organisms that have been altered or produced through recombinant DNA techniques, and both of these conditions have to be met if that GE organism could be a plant pest or have characteristics. So some of what triggers that, as Sarah mentioned, is if the donor organism or the recipient organism or the vector used to transform the organism is a plant pest. And plant pests are defined by statute as organisms that can directly or indirectly injure, cause damage to, or cause disease in or to any plant or plant products. And there's a whole list of things listed in statute that fit that. Sarah also mentioned the AMI-regulated process we have. I won't spend too much time on this, only to say that in the last few years we've instituted a process whereby developers can send an inquiry to us as to whether their particular GE organism meets the definition of a regulated article. When they submit a letter of inquiry to us, they should be inquiring about an actual organism, not some hypothetical thing that they may or may not make in the future. And we provide instructions for submitting inquiries on our website. After we respond, both the inquiry and the response are posted on the website, and since July of 2011, we've responded to 34 such inquiries. What we regulate, if you do have a regulated article, we'll regulate the following activities: importation into the U.S., interstate movement within the U.S., or environmental release and field tests. And we do this through 2 different ways. We have what we call permits and we have notifications. And the permit notification procedures are used to authorize the activity that's been proposed, whether it's an importation, a movement, or environmental release. I'll point out here that any unauthorized importation, interstate movement, or release of a regulated article is a violation of the regulations, and that would include accidental releases. So if you're working with a regulated article, something that would be a regulated article in your lab and it gets out, that would be considered an accidental release and that would be a violation of the regulations. I'll quickly summarize some of the differences between notifications and permits. Basically, the notification process that we have is an administratively streamlined option. It is available only for genetically engineered plants, not for any other genetically engineered organisms that fall into our authority. Those plants must meet 6 eligibility criteria, which are specified in our regulations. In general, notifications require less information to be submitted to us, and our response procedure is faster. Some of the other key differences between notifications and permits are that notifications are only valid for 1 year, whereas permits can be valid for 1 year or for multiple years. Right now, we can issue permits that we're issuing permits that extend up to 3 years in time. Notifications have to meet releases and movements and so on that fall under notification have to meet a list of performance standards, which are also specified in regulation, and I'll talk about those briefly in a minute. Whereas in permits, we will actually specify specific standard and supplemental permit conditions. And notifications can't be amended, so you can't add things to a notification later, whereas permits can be amended later. I'll briefly go over the notification criteria for GE plants. I'm not going to go through all of them, but basically they include that the recipient, the organism, so the plant can't be listed as a noxious weed or considered by APHIS to be a weed in the area where it's going to be released. And we do have a list of noxious weeds under a different part of our regulations at 7 CFR Part 360. The genetic material has to be stably integrated into the organism, and its function needs to be known, and it can't result in plant disease. And then we have a number of more specific restrictions on specific introduced genetic material that's not available for the notification procedure, such as material that would encode products intended for pharmaceutical or industrial use, coding sequences whose products are known or likely to causal agents of disease in humans or non-target organisms, and others, all of which are specified in our regulations. I mentioned that we have also 6 performance standards. I think the most important with respect to the deliberations of your committee in thinking about the release of gene drives is that field trials must be conducted such that the regulated article will not persist in the environment and no offspring can be produced that can persist in the environment. And upon termination of the field trial, any volunteers, and again this is plants, so we're talking about volunteer plants, shall be managed to prevent persistence in the environment. So I think you can see from this that any insects that would fall under our regulatory authority that were engineered to contain gene drives would, of course, not be eligible for notification. And similarly, I think many of the plants that have been discussed, for example, engineering herbicide susceptibilities into weedy species, would also not be eligible for the notification procedure. For permits, we have, as I said, standard permit conditions and supplemental permit conditions. The standard permit conditions are placed on both movement and release permits to minimize the possibility that GE organisms will persist in the environment or produce offspring that persist in the environment or significantly impact non-target organisms. And we add supplemental permit conditions to regulated field trials in order to maximize confinement. And these are generally customized to the particular organism trait and release locations. So some of our typical supplemental permit conditions include requiring a minimum separation distance to fields of the cultivated crop so that we don't, to try to minimize gene flow from the regulated article to sexually compatible relatives that are other cultivars of the crop. Also minimum separation distances to wild relatives that are in the environment. buffer zones around the regulated field trial, and then follow crop planting restrictions and post-harvest monitoring requirements that specify things such as how frequently you have to monitor, how long you have to monitor, and methods for devitalizing any volunteers that occur. We place stricter conditions on regulated field trials of GE plants producing industrial or pharmaceutical substances. So, to give you a sense of the scale that we're talking about, this is the number of authorized field release sites that we, the number of field release sites authorized each year since 1989, and you can see that in 2014 we were approaching 11,000 field release sites authorized, 120,000 different authorized constructs. All that is actually a little bit of a misnomer because of the way these numbers are counted. This is counting at sometimes specific elements within a larger construct, so probably a more accurate number here would be around 40 to 50,000. But it's a very large volume of organisms of GE plants that we are processing every year and authorizing for imports, movements, or releases. For plant pests, insects, we've had a much lower volume, the kind of thing you can almost count on two hands. We've had 16 authorized plant pest insect movements. I should say, this should say GE insect plant pests. We do have many movements of microbes and so on, but on the insects, we've had 16 authorized movements: field cricket, pink bollworm, diamondback moth, and Mexican fruit fly. You'll see from this that most of them simply contain visual markers or are the repressible dominant lethality systems that cause female lethality. As Sarah mentioned, we conduct a facility inspection prior to authorizing these types of movements. We look particularly at the structure and siting, at the equipment in the facility and how the facility operates, things like SOPs and so on. Much of this instruction follows the guidance documents that Sarah mentioned earlier. And we work with plant protection and quarantine, APHIS plant protection and quarantine for this purpose. In addition to movements, we've had several GE insect plant pest releases since 2001, 15 authorized releases of GE pink bollworm, and I'll point out here that 14 of those releases were sterile insects with visual markers. One release of conditional repressible lethal insects, and that was a caged trial. We did complete environmental assessments prior to authorizing these releases back in 2002 and 2006. And we have completed an environmental impact statement for a large-scale release back in 2008. It also addressed medfly and Mexican and Mediterranean fruit flies, although a large-scale release of these GE organisms hasn't occurred yet. Recently, we also authorized the release of genetically engineered diamondback moth. This is, again, a conditional repressible lethal system causing female lethality. Confinement is essentially achieved via climate. So this is occurring in upstate New York, and when the winter months come on, the organisms die. And we completed an environmental assessment prior to authorizing this release. We don't, I should say, we don't complete environmental assessments or environmental impact statements before most of the releases we authorize. We do an analysis to see whether most of our releases or movements are conditionally exempt from the NEPA requirements, and only if they're not conditionally exempt will we conduct an environmental assessment. But these are examples where we could not conclude that they were conditionally exempt, and so we conducted the environmental assessment. I want to touch briefly on the difference between containment and confinement as we view it. So containment procedures are procedures used to prevent exposure of GE organisms to the environment. This generally refers to use in labs, in greenhouses, and during transport. And here what we're really aiming at is a probability of release near zero. Confinement, on the other hand, for us means procedures used during field trials to ensure that the organism does not persist in the environment. So these include some of the reproductive isolation and post-harvest monitoring requirements that I discussed earlier. And here what we're aiming at is a probability of persistence that should be near zero. For confined field trials of GE plants, we have several risk considerations as we go through an analysis. We've identified possible hazards in 4 broad categories: the potential of the plant to become a weed of agriculture or be invasive of natural habitats, the potential for gene flow from the plant to sexually compatible relatives whose hybrid offspring may become more weedy or more invasive, the potential for the plant to itself become a plant pest, or the potential impact of the GE plant on non-target species. And basically, for confined field trials, because, you know, we're as you've seen, we are reviewing so many thousands every year. A complete environmental risk assessment simply isn't possible, both because of the sheer number, but also because the data required for a complete environmental risk assessment may not yet have been generated. Many of our applicants are, especially the companies, are doing large-scale screens for desirable traits, and part of their screening process requires that they be tested in the environment to see how they behave in the environment. And this occurs, you know, and they down select from the results of there. So much of the data required hasn't yet been generated, and the full extent of potential hazards can't necessarily be known. So that's why our goal really is to minimize exposure in order to minimize risk, because if there is no exposure, then the risk is zero by definition. And as I mentioned, in some cases, approval of confined field trials requires a complete environmental risk assessment. So in addition to the ones I've mentioned and before, also if we have a new species that's never been subject to a release before, or industrial GE crops and plant pests, as I've mentioned. In addition to all of this upfront regulatory work, we have an active program of compliance inspection to verify that people holding notifications or permits are complying with our regulations. All field trial sites are eligible for inspection. We require timely submission of planting reports that detail what was planted, where it was planted, any unexpected events that occurred or observations. Applicants must allow us access to field sites, and we also achieve compliance through a variety of other measures, including, in addition to inspections, we have educational and outreach efforts and various audits. We do take corrective action in the case of a non-compliance event, and in some cases we may refer a non-compliance event to our Investigative and Enforcement Services Division of APHIS for formal investigation. Failure to comply can result in increased regulatory oversight, and in the most serious cases, pretty large penalties and fines. So unlike FDA, we don't do what Larissa talked about, needing a pre-market approval. We don't do that for things that are going to be taken to market. Instead, what happens, and this is with GE plant development, is that typically the developer will start with laboratory and greenhouse studies, and these are mostly not regulated by APHIS. We would essentially regulate movements of the materials to the laboratories and greenhouses if it's an import or an interstate movement. They then progress to field testing, which, as I've discussed, is regulated, and then those products that a developer has decided that they want to commercialize would not be, commercialization itself is not regulated by APHIS. Instead, what happens in that case is we have a procedure whereby regulated parties can petition us for non-regulated status for their GE organism. And if we approve their petition for non-regulated status, then that organism would no longer be subject to the regulations under 7 CFR Part 340. When we review a petition, we base our decisions and actions and recommendations on the best science available. Essentially, the developers must demonstrate that their organism does not pose a plant pest risk, and like FDA, we require information such as molecular characterization data, phenotypic characterization, and observations of how the organism is behaving in the environment, and so on. We also have a public review process whereby the public can review the petition and our evaluation before and have the ability to comment on those documents prior to our final determination. Our process, in our petition evaluation process, we do 2 evaluations. First is a plant pest risk assessment to determine if the GE organism is a plant pest, and that tiers back to our actual regulatory authority under the Plant Protection Act. We also do an environmental assessment to evaluate the potential environmental impacts arising from APHIS decision, and that tiers back to NEPA. Larissa gave a very good overview of NEPA, so I won't go into that. The only point I will make is that ultimately our decision is based on the first risk assessment. NEPA does not give us any authority to approve or deny a petition for non-regulated status. It only enables us, it only informs our considerations. The ultimate decision is based on our plant pest risk assessment. The only exception to that would be, I suppose, if USDA itself is intending to undertake an action, in which case the NEPA analysis might inform USDA's decision about whether to actually undertake that action or not. But again, if another part of USDA petitioned us to determine if the GE organism can have non-regulated status, our decision in APHIS would be based on the plant pest risk assessment. To date, we've made 100 determinations of non-regulated status in response to 117 products, all plants, from 17 plant species listed here. Our determination does extend to the GE plant and all of its offspring. And as I said, once we determine that an organism has non-regulated status, that organism can be freely moved and planted without APHIS regulatory oversight. So we don't have a system of post-approval reporting in our system. In the past, most of our determinations of non-regulated status have been event-based, similar to the approach that Larissa described. We still take an event-based approach, although there are some cases where we might see if we can begin to move away from that. At the moment, we have been more actively using an extension process for determinations of non-regulated status, whereby a developer can apply for an extension of a previous determination of non-regulated status to a new organism based on similarity to the previously deregulated organism. This just gives a quick list of the plants that have received non-regulated status, the 17 plant species. In green, you see the ones that are actually, green and yellow shows those plants that we know are in commercial production, and the various types of phenotypes that have been conferred on the plant through genetic engineering are listed as well. Finally, and this will be my last slide, we do have guidance documents available for the research community, the developer community, and so on. We have users' guides for how to apply for notifications and permits and what information to include. We have a list of minimum recommended separation distances for 9 plant species: alfalfa, canola, corn, cotton, rice, safflower, soybean, tomato, and wheat. And we have guidance for submitting petitions, including the types of data and information to collect and include. But we very much encourage parties that are interested in submitting a petition to consult with us well in advance of submitting the petition to describe what it is that they'll be submitting a petition for and to talk with us about what specific types of data we might be interested in. In addition, as Sarah mentioned, Plant Protection and Quarantine has containment guidance documents for arthropods and noxious weeds that cover construction, equipment, and operational standards, and we refer to those. And so with that, I will end. This just tells you where you can find more information about what we do, and I guess now the presentations are done.